This video we are going to discuss about cloning vector based on viral DNA. Now this viral vectors are those vectors in which the gene of interest is incorporated into a virus. Okay, the gene of interest is incorporated into the genome of a virus. Okay, that thing is your viral vector. Now the thing is that the virus infects cells at high efficiency. Okay, viruses are highly efficient in terms of infecting a cell, and that is the reason why if you use virus vectors, then cloned genes can be introduced in a particular cell at a higher frequency than simple transformation. Okay, so we use this property of virus, this ability of virus to infect cells at a high efficiency to introduce the cloned genes at a higher frequency into a particular cell. Now, some viral vectors are specialized for producing high levels of proteins. Those proteins were encoded by the cloned genes. Okay, so you have cloned genes in your viral vector, and then this viral vector, with the help of that, they produce high levels of proteins in your host organism. Now, this M113 based vectors, these are the type of viral vectors which facilitate sequencing, and they also facilitate the generation of mutations in the cloned genes. Okay, so for sequencing and for generation of mutations in the cloned genes, you can use M13 based vectors, which are the type of viral vectors. Now let's look at the cloning vector based on lambda phage. Now this lambda phage is a template phage and it infects E. coli. And this lambda phage replicate by lytic pathway as well as lysogenic pathway. Okay, it can show lytic pathway or lysogenic pathway. Now the lambda phage genome consists of a single linear double-stranded DNA whose size is about 49 kb. Okay, so the genome of lambda phage is just of about 49 kb in size. In the lambda phage genome, okay, in this single linear double-stranded DNA, okay, in this molecule, at either end there is a short 12 nucleotide stretch of DNA, and this short 12 nucleotide stretch DNA, which is present at the either end of this DNA molecule of lambda phage, is single stranded, and it is described as cohesive end or sticky ends. Okay, so if you consider the DNA molecule of the lambda phage, then at both the ends of that DNA molecule there are cohesive ends or sticky ends. Now this lambda cohesive ends, which we just saw, are known as cos ends. Okay, cos ends are just cohesive ends, and they have two roles. Okay, they play two roles in the lambda infection cycle. The first role is that these cohesive ends allow the linear DNA molecule which is injected in the cell to be circularized. Okay, the linear DNA molecule is able to form a circle because of these cohesive ends. Now the second role of cos sites is rather different, and it is there in the lytic cycle. Now during lytic cycle, a large number of new lambda DNA molecules are being produced by rolling circle mechanism of replication. Okay, by doing rolling circle mechanism of replication, a large number of new lambda DNA molecules are produced during lytic cycle. Now this results in the formation of this catenate, and that this catenate consists of a series of linear lambda genomes which are joined together by cos sites. Okay, catenate means there is this long molecule, long DNA molecule, and these are the cos sites. Okay, so what will happen is that this molecule will be broken down. Okay, and then this individual genomes will be packed into your viral proteins, and that is how the new virus will be produced. Okay, so as the virus replicates at a faster rate, so it is not going to synthesize genome one by one. Okay, it just synthesizes a long DNA molecule, okay, which has cos sites in them, and when these cos sites are subjected to cleavage, then that results into individual genome, and these all are identical to each other. That's the point. Okay, so yeah, that's the function of these cos sites. Okay, so to make this thing possible. Okay, so the role of cos site is now to act as the recognition sequence for a particular endonuclease. Okay, this endonuclease will cleave the catenate at the cos sites, and this cleavage of the catenate at the cos sites will produce individual lambda genomes. Okay, so yeah, so the cos sites act as the recognition sequences for endonucleases. In lytic pathway, the viral functions are fully expressed. Okay, in lytic pathway, the viral DNA and proteins are quickly produced, and they are packaged to produce virus particles, and then there is lysis of the host cells, which then results in the release of the viral particles and virions. Okay, viral particles or virions. Okay, so that is how it is. In lytic pathway, the viral DNA and proteins are quickly produced, they are packaged, then there is lysis of host cell, and then there is a release of these viral particles. But like in lysogenic pathway is not so quick. Okay, in lysogenic pathway, what happens is the phage DNA gets integrated into the host cell genome, and then this phage DNA replicates together with the host cell DNA for many generations. And in this time, okay, when this phage DNA is integrated into the host cell DNA, the phage DNA is inactive. It does not show any activity. So the host is kind of working normal. But in certain environmental changes, okay, if certain environmental changes happens, then that trigger, okay, those certain environmental changes trigger the expression of that dormant viral DNA. And once it is expressed, then there is formation of progeny of viruses, and then there is lysis of host cell. Okay, so it takes a quite longer time for lysogenic pathway. Okay, for like by production by lysogenic pathway will take considerably large amount of time as compared to lytic cycle. Now, one problem that we face while using the lambda phage DNA as a cloning vector is its size limitation. Okay, lambda phage DNA is kind of very small. Okay, so the normal lambda phage genome size is about 49 kb. Okay, and the capacity of the capsid to incorporate the maximum genome size is 53 kb. Okay, what is capsid? Okay, so this is your DNA which is produced in the host cell. Okay, this is your viral DNA. Okay, now capsid is that cover which will encapsulate this viral DNA, and that is how your virus particle will be produced. Now the maximum capacity of this capsid to hold the DNA is 53 kb, and the size of this virus genome is 49 kb. Okay, so you just get 4 kb of the 4. This means you can only incorporate the DNA, target DNA up to 4 kb. Okay, so the lambda phage DNA molecule can be increased in size by addition of only 3 kb new DNA molecule. So your target DNA size can be only 3 kb, and that is a quite small size. That's the problem while using viruses as the cloning vectors. Now that problem of 3 kb can be overcome by deletion of non-essential DNA. Okay, there is non-essential DNA on your viral in your viral genome, and if you delete that non-essential DNA, then you will be having a space, and the non-essential DNA present in a viral genome, lambda phage genome, is about 15 kb. So when you delete 15 kb, you have 3 kb, so you can insert 18 kb of new DNA. Okay, you can clone 18 kb of new DNA using that lambda phage vector, cloning vector. Okay, so the non-essential region that we just deleted were the genes responsible for lysogenic pathway. Okay, so the deleted lambda phage genome will be now non-lysogenic. That is, it will only follow lytic cycle. It will not follow lysogenic cycle, and that is a good thing for the experiment. Now there are two types of this lambda vectors. Okay, the first type is insertion lambda vectors, and the second type is replacement lambda vectors. Now this insertion lambda vectors are the simplest form of the lambda vectors. Now during this development of this insertion vector, what happens is the large segment of the non-essential regions of the lambda phage genome is deleted, and then the two fragments are ligated together. Okay, there is your non-essential region on the lambda phage genome. Okay, that is deleted, and whatever the gap was produced, those two fragments which were produced because of this, they are ligated together. Now this lambda phage genome contains at least one unique restriction site, okay, for the insertion of the new DNA. Okay, insertion lambda vector have at least one unique restriction site for the introduction of new DNA. Okay, then you will cut that genome and you will add new DNA, and that is how you are going to incorporate new DNA into this insertion lambda vector. Now the size of the insert, okay, the size of the DNA fragment that an insertion vector can carry depends upon the extent up to which the non-essential DNA is deleted. Okay, how much non-essential DNA you have deleted, on that the carrying capacity of this insertion vector will depend, and normally the carrying capacity of this insertion vector is about 8 to 12 kb. Okay, so the insert of DNA fragment of size about 8 to 12 kb can be carried by this insertion vector. Now let's talk about a replacement vector, the other type of the lambda vector. Now the loading capacity of this replacement vector is about 10 to 23 kb. Okay, this replacement vector is a full length lambda molecule, and it has two identical restriction sites flanking the non-essential region, which is known as stuffer fragment. Okay, so the stuffer fragment which is there in the genome of this replacement lambda vector, that stuffer fragment is flanked by two identical restriction sites. Now what happens is this non-essential stuffer fragment is deleted and replaced by the foreign DNA, and that is how the target DNA is inserted in your replacement lambda vector. Okay, the stuffer fragment is deleted and it is replaced by foreign DNA. Now The central segment of this lambda genome consists of genes which are responsible for lysogenic cycle, and that is the reason why these genes are not essential for lytic cycle. Okay, the purpose of your experiment is that you should be able to insert the target molecule into this vector, and this lambda vector should show the lytic cycle. Okay, then you will get the experiment done. Okay, so the genes which are responsible for lysogenic cycle are not useful. Okay, they are not essential for the experiment, and that is the reason why these genes responsible for lysogenic cycle, which are present at the center of your lambda genome, are removed and replaced by the foreign DNA fragment. Okay, so that is how this replacement lambda vector works. Okay, so it is possible to clone a foreign DNA of about 23 kb using replacement lambda vector, and that is the reason why these replacement vectors are used in genome libraries. Okay, in genome libraries you use replacement lambda vectors. Now let's look at the insertion vector. Okay, so this is your double-stranded DNA and it is of about 40 kb. Now there is a restriction site. So you cleave that restriction site, you take new DNA and you ligate it on that, and that is how your new DNA molecule is produced, okay, which is of 52 kb, and this thing is your insertion lambda vector. Okay, this white part is the lambda vector lambda genome, and this blue is the new DNA, that the target DNA that you want to replicate. Now this is how the replacement vector works. Okay, you have this lambda genome here, and in between there is this stuffer fragment which is not essential. Okay, so and it has two restriction sites, two identical restriction sites flank this stuffer fragment. Okay, so by using restriction enzyme you cut this stuffer fragment from this lambda genome, and then there is a gap. Okay, and then you fill that gap, you ligate that gap with your target DNA, with your insert, and that is how the replacement lambda vector is able to carry your target gene now. Okay, so originally it had a size of about 41 kb, then you removed stuffer fragment, then it got about 30 kb, and then you can insert up to 23 kb now. Okay, so its size can be this final vector molecule may be having size of about 53 kb. Now let's discuss about in vitro packaging. So what is this all about? Okay, so here is the thing. We produced this recombinant DNA molecule. Now we want to replicate this. Okay, so we should be able to wrap up this DNA molecule into the viral coat, okay, so that it can infect the bacterial cell and it can replicate there. Okay, that is how your experiment is going to be successful. Now you have produced this recombinant DNA molecule by combining the viral genome and by combining your target DNA. But now you have to pack this viral genome into the viral coat. Okay, now how are you going to do? That we are going to discuss in this in vitro packaging. Now this packaging requires a number of different proteins which are encoded by the lambda genome. Now these proteins required for this in vitro packaging can be prepared in high concentration from the cells which are infected by defective lambda phage strain. Okay, so to produce this packaging proteins, you use defective lambda phage strains. Okay, so for doing that, two different systems are used. Now let's see about those systems. Now starting with single strain system. Okay, what happens in this system? Okay, in this system the defective lambda phage is there. Now this defective lambda phage is carrying a mutation at the cos sites. Okay, so at cos sites there are mutations. So these cos sites will not be recognized by the endonucleases, and that is the reason why there will be no cleavage of the catenons, which is very essential for the phage replication. Okay, so as the phage produces this long DNA, okay, this long DNA molecule is produced, which is known as catenons, and there are this cos sites in it. Okay, after the endonucleases cuts these cos sites, there are this individual genome, viral genome, which then they are packed with the proteins, and that is how your viral DNA is produced. But if this long DNA molecule, if this long catenon is not cut by the endonucleases because there is mutation in the cos site, then the individual genome of the viruses will not be produced. Okay, and there will be nothing for the proteins to coat, and that is how you will get the proteins, and that proteins you can use for your own experiment. Okay, so that is how you obtain those in vitro packaging protein, by just doing mutation at the cos sites. Okay, so this defective phage genome cannot be packed by the capsid. Okay, so there is direct synthesis of all the proteins which are required for packaging, and these proteins which are required for packaging are not used because the phage genome is defective now. So the proteins accumulate in the bacterium, and you can purify those proteins from the cultures of E. coli, which is infected by that mutated lambda phage. Okay, which had the mutation at the cos sites. Okay, so you infect E. coli with this mutated lambda, then you purify the proteins, the packaging proteins from it, and then those proteins you will be using to package your own recombinant viral DNA molecule. Okay, so those protein preparations are then used in vitro packaging of the recombinant lambda molecules. Now the second system by which you can obtain those packaging proteins is by two-strain system. Okay, so in this system we are using two different defective lambda strains. Okay, you are taking two strains and both of them are defective. Now this both of strain contains a mutation in that gene which is responsible for the production of one of the component of the phage protein coat. Okay, so one strain is having a mutation at gene D, the second strain is having a mutation at gene E, and both of these genes code for protein which is required for phage coat. Now what will happen is that both of these strains, which are grown in separate colonies, will not be able to complete lytic cycle in the E. coli. Why? Because they have absence of a product of the mutated gene. Okay, the mutated gene will not give the product that was essential for the formation of this phage protein coat. So the phage protein coat will not be formed, and that is how the lytic cycle will not be completed. Okay, so that is the reason why the complete capsid structure cannot be produced by both these strains. Okay, so what for in vitro packaging, what we do is we prepare a mixture, okay, combining the lysates of these two cell cultures, one of which had the mutant D strain and the other had the mutant E strain. Now that mixture will be having all the necessary components for in vitro packaging. Okay, so each lysate that we mix has the phage tails, had the assembly proteins, and had the components of the head except D or E. So this is what is happening. Okay, as we said that one strain was having mutation at gene E and other strain was having mutation at gene D. Okay, so if gene E is mutated, so E protein will not be produced. Okay, and that is how the protein coat will not be formed, because E is essential for that. Okay, so protein coat will not form. And considering GD, if in the second strain the gene D is mutated, and that is the reason why D protein will also not form in that culture, and again the protein coat will not form because this D protein was also essential. But the thing is that after making culture of both of these, okay, you add them, okay, you mix them, and now in the solution you have E proteins, you have D proteins, and you have all the other necessary proteins which will be required for the formation of the capsid. And that is how now your recombinant DNA you will put in this mixture, and that is how your recombinant DNA now will be packaged in vitro with the help of all these proteins. Okay, so although the mixing is done in vitro, the components will self-assemble and will produce a functional phage that can infect E. coli. So that's all for this video. Thanks for watching.